It's Miss Parrott. We're going to get ready for the Marine Science, the AICE Marine Science Paper 4 exam. And first we're going to do a practice from the October-November 2023 Paper 4-1 test. You can get a blank copy just by Google searching that, or in the description below, I'll post a blank version of it through my Google Drive. We're going to need some highlighters, a pen for our written answers, a pencil. I always go with Ticonderoga. Ticonderoga has the best erasing ability despite its pink top, and we're going to need it for any diagrams, any data tables, any graphs, or any biological drawings. A ruler, because all of our lines need to be straight except in your biological drawing, and a calculator. Let's get started. All right, we're going to get started. Make it a little thicker. Great, okay. We're only going to use a black or dark blue pen, any HB pencil, which is why I recommend Ticonderoga for any diagrams, graphs, or biological drawings. Do not use erasable pen or any whiteout. Don't write on the barcodes, which seems obvious, like who's going to write on that. And you may use a calculator. You absolutely should use a calculator. Though you still have to show your work, like what you'd be typing in, you absolutely should be using a calculator. Show all of your work, even if they're not asking for it. If they are asking for it, you are being graded on that, and definitely use appropriate units. All right, our first question is about the desalination plant. Industrial factories that produce fresh water from seawater, and they are thought to be be harmful to seagrass beds due to the release of highly saline water into the sea, and we refer to that as brine. And you see a cell of a seagrass leaf. You can tell that this is a plant cell because it has a cell wall. It also has a chloroplast, and it has a large permanent vacuole, which is actually what this area X is. This here, it is also referred to in the inside is called a tonoplast, but it has dissolved solutes. It also has sugars in there. It has can hold some waste. It also has water, and it helps add towards the turgor pressure of the plant cell. Name the organelle labeled X, there we go, and state its function. This is the large permanent vacuole. Its function. Storage for nutrients. And also referred to as cell sap. Okay, we have some magnification. The diagram has a magnification of 30,000. Calculate the actual width of the seagrass cell between A and B on the diagram. Show your working and state the unit. Okay, so this is where we got our triangle math at. Magnification is the image, what the image measures to be divided by its actual. We want to find A. They said calculate the actual width, so we need to be looking at what is the image divided by the amount that it's magnified. So we're going to need to get our ruler. And something I just want to put out there, it might just be the way that this is positioned on the iPad, like take up its full space. But measuring it on here is actually not the same as what I was getting when I was doing it on the printed test. Okay, so I'm going to start it here where we're at one edge of A, and then B. So I'm getting about 5.1 millimeters. When I did this on the printed version of this, I got, oh, sorry, it's not 5.1. 51. When I did this on the printed version, I had 47 millimeters. So whatever you're measuring is, you know, as long as you show it that way, and again, I think it's just the way that this is oriented on the iPad. No, it shouldn't be. Anyways, make sure you show what your measurement is on there. So I'm going to actually run with this as if it was 51, but again, when I did it on the paper, I measured it to be 47. Okay. First thing I'm going to need to do is convert. And in 1 millimeter, because I need these to cancel, they have 1,000 micrometers. Millimeters are going to cancel, which is great because I need to be left with the actual unit for measuring things that are cellular size. 51 times 1,000, you're going to get 51,000. Micrometers. And then we're going to divide it by how much it's magnified by. It was magnified by 30,000 times. Okay, and you should get 1.7, and we need to state the unit, micrometers. You do get a mark to show that. So the 51 times 1,000, you get another mark to show this entirety that you're dividing it. And then the last mark is that you have the answer with the unit. If you did it on the printed paper and you measured it to be 47, then you would be doing 47,000 divided by 30,000, and you would get 1.57 for that. So just make sure no matter what you're showing your work so that they can go back through and see where you're getting these results from. All right, scientists investigated the possible impact of desalination plants on seagrasses. They grew seagrasses in water of different salinities, your independent variable, and they measured two things. They measured the percent of water inside the seagrass cells and then the water potential of the seagrass cells. The percentage of water content of the seagrass cells and the water potential of the seagrass cells were measured from the start, after 4 weeks, and then after 6 weeks. Water potential was measured in kilopascals, or kPa. And so we see the results. All right, in our graph, just looking at this first one, figure 1.3, you have the percentage of water content in the cells. The different bars are showing the different times that they took the measurement. All right, and typically I like to just try and make sense of this first before I actually read the question for it. But things that I am noticing really quick is at 35 parts per thousand between the start, 4 weeks and 6 weeks, we really don't have much change. like little to no change. And I use the delta symbol to show change in things, just instead of writing it every time, I just will use that as like a quick note. So this triangle is the symbol delta, and that means change in. So there's little to no change. 37, very similar. It loses like 2% right here between that beginning and the 4th week, and then we go up 1% pretty much. There's really not much change. Same thing. Not much change. 46, we go down 2%, and then we stay the same between week 4 and week 6. So same, there's not much change. And this is in the water inside of the cell. Thinking that as salinity is going to increase, that's going to cause the solution to become more hypertonic, and these cells will start to lose water. So we're kind of looking at what is the higher salinity that's going to cause this. At 54, we start to see some bigger changes. So initially, our cell has, if we look here, 1, 2, 3, 4, 5. We're jumping up by 10 with 5 lines in between. It means each box essentially is going to be equal to 2. And so if I'm looking right here, it's going to be 78%. And then after week 4, we go down just about 68%. So we lose 10% of the water. And then at week 6, we're just at about 62. So comparatively, there is much more water loss in 54 parts per thousand. All right, so they're losing water between the start of this and then 6 weeks. And I imagine this will probably be something we're going to have to answer, but the 78 to 62, they lost 16% within that 6-week period. Okay, and then the water potential. And this is the water potential of the seagrass cells. At 0 kilopascals or 0 kPa water potential, this is where water is like 100% fresh. So this negative 6,000 is going to be where there's the highest salinity. Okay, same thing. We're going to be looking at this between 35, 37, 46, and 54 parts per thousand. And again, the shaded bars will show you over those different weeks. 35 is our average salinity. There's really not too much difference between them. We start to see that the water potential is going to go down, but then it kind of levels itself out. We see a very similar trend that we did in figure 1.3, where there's not a ton of change. 37 to 46 parts per thousand. Like this week 4 right here, and week 4 at the 46 parts per thousand, those are not really much different. Same with the week 6, there's really not much difference. But at 54, we have a pretty big difference. A pretty big difference. So we know from figure 1.3 that they're losing water. But we start to see that their water potential is going down. And so when water potential goes down, that means, like, we wouldn't want to answer this way, but a way to think of it is that whatever fluid inside of the cell is there, it's not being composed mostly of fresh water. So if a way to think of it is like the cell itself is getting saltier, or it's taking on some of that salt water. We start to see that happen between the beginning and then week 4 to week 6. The salt in the cells is increasing. Just because, water will behave a certain way. Water goes from a high water potential to a low water potential because of osmosis. But salt will also, diffuse through facilitated diffusion. All right, and then I feel comfortable with that. I just like to try and make sense of kind of what I'm looking at. So the 35, 37, and 46 really doesn't have a big difference, right? These plants are adapted to be in high salinity areas, but then there is a limit. Use the data. Oh, I don't know what this number is. Don't know the numbers. Use the data in figure 1.3 to describe the effect of growing the seagrass in water of different salinities. Figure 1.3 is this first one, looking at the percentage of water in cells. Okay, and so we said there was little to no change. That's fine. Okay. When I do a bullet point, that typically just means like this is a marking point. Instead of me putting what the specific marking points are, I'm just going to denote that with different bullet points. So there's little to no change in the percent of water between 35, 37, and 46. Okay, and obviously we're not going to answer like this on a paper 4 or a paper 3 or anything like that. Sorry. We're not going to put just a triangle for the change in. We're not going to put, you could probably put the percent symbol, but you could also just write it out as well. So I'm just writing it shorthand just for the sake of practice with us. The percent of water decreases at 54 parts per thousand. Something that we need to look at, though, is they're showing us these error bars. So this is the standard error using confidence intervals. Things we want to look for is if there's overlap. So at 34, 37, and 46, where we say there's not really much change, not only that, but we can see that there's overlap between these error bars. So, initially, maybe in AS level, we just looked at just the bar itself, right? So we would just be looking right here, and we're like, oh, yay, look, the average loss is going to be 78%. But then once we actually calculate the standard deviation and then the standard error and its confidence intervals, we actually can start to see that it could be 80 or it could be 76. That is going to be like the range that these means can be in error. And it does overlap. So where my bottom limit is right here, let's actually make that a little bit smaller. it does overlap with my 4-week bar. And likewise, my top error, the top part of the error bar also has some overlap too. So they're not really significantly different from each other. Again, just looking at the bar itself, it might seem that way, but when we look at the error bars applied, it's not necessarily the case. I'll do the same over here, and I'll try and use this one. So here, the bottom limit of this, we do have overlap with the 4-week and the 6-week at the 46 parts per thousand. Now the top limit isn't really overlapping, but the top limit of my 4-week and 6-week overlaps with the start. So their averages really are not significant from each other. Now 54, we don't see this pattern. My top limit isn't really overlapping anywhere with the initial. My bottom limit is not overlapping with of the 4-week with the 6-week measurement. Likewise, this doesn't have any overlap either, my 6-week measurement. So they are different from each other. But back to what we were talking about with the 35, 37, and 46, we do have overlap of those error bars. So those salinity differences didn't really have that big of an effect on them. At 54, we can see that from the initial to week 4 and then to week 6, we do have a decrease over time. .54 parts per thousand. There's a continued decrease in percent of water in the cells over time. And then we can also give some data proof. So I tell my students all the time, if you're given a data table or you're given a graph, use those values to support your assessment or your description of the effect of whatever they're asking you. And so the main difference that we're talking about here is what's going on at 54 parts per thousand. Likewise, you could compare that there really wasn't much of a salinity change in either the 35, 37, or 46. You can talk about that. I don't want to see this. Cool, cute, great, awesome. Okay, and so this is where, if I'm saying it's decreasing over time, I think the easiest thing is just to subtract. So like I did here, the 78 to 62. Just looking at the means. At 54 parts per thousand between the start and 6 weeks of the experiment, the percent of water decreased by 16%. Actually, I'm going to put it back up there. How annoying. Okay, and this is only 3 marks. Highlight your brackets. Okay, now use the data in both figure 1.3 and figure 1.4 explain. So give reasons for the changes in water potential over time when the seagrass is placed into water 54 parts per thousand salinity. So isolating just 54 parts per thousand, they want you to give reasons for the difference in water potential. And we need to use both. So obviously 54, we see that there's more water loss inside of the cells, and then their water potential starts to decrease inside the cell. All right. Give reasons for. So the water potential is going down for a couple of reasons. One, we're losing fresh water, and the salts inside of the cell are starting to increase. So water, and if you're wondering why am I putting H2O versus just writing out water, there's literally no reason. Water's leaving the cell. Why? Because we're doing explanations. So the reason why is due to osmosis. Why? So all these are why? Explain, explain, explain. The water potential was higher on the outside of the cell. Now you could also speak of it in terms of the reverse argument. You could say the water potential was lower on the inside of the cell. That doesn't make sense. Hold on. And you know I don't really ever edit my videos, so we're just going to live with me. That doesn't make sense. Because water goes from a high water potential to a low water potential, and water is leaving. So the water potential was higher on the inside of the cell. That makes sense. Move up here real quick. And so in class, I usually do this. So we're going to have a high water potential, which is shown by like a pitchfork that is like the symbol for water potential. Outside of the cell, we'll have a lower water potential from all that brine. And then I compare the salt concentrations too. The salt is going to be higher, the dissolved solids, and inside the cell we're going to have lower dissolved solids. And water is going to flow from a high concentration, which is inside the cell, to a lower concentration, which is outside the cell. It's going to flow out. And then this cell actually can start to lose its rigidity and its turgor pressure. And your cell membrane can start to pull off from the cell wall, which is called being plasmolysed. Okay, so the water potential was high on the outside of the cell. I'm sorry, I said it again. On the inside of the cell, just like I wrote. Over time, the concentration of salt inside the cell starts to increase, and we start to see that between the initial to week 4 and then again to week 6, because it's clearly not gaining fresh water. inside the cell increases. And the arrows, I use arrows a lot whenever I'm practicing exam questions with students. We're not going to use arrows on your exam. Okay, which causes the water potential to further decrease. And scroll back up one more time. And this is why we're losing throughout these this entire experiment. So the start to 4 to 6 weeks. So that's the reason why we're continually losing water inside of them. To be lost over time. All right, and then another 3 marks. Desalination plants also release toxic heavy metal ions into the seawater, and it's toxic. So give reasons why, explain. This is always your why. The release of heavy metal ions is harmful for top predators in the ecosystem. And so this is going to be a question where we need to talk about bioaccumulation and then biomagnification. So within your lower trophic levels, the toxin levels might not be that high, but as those organisms get consumed by higher trophic levels, it starts to magnify up those trophic levels. And we find that it ends up looking something like this. Okay, where down here is like your producer or maybe even like a filter feeder, your lower trophic levels. They accumulate in their tissues, and then organisms that consume them, they're going to have it accumulated in their tissues, and then organisms that consume them will have it accumulated in their tissues, and this continues. And what you see on a large scale, though, if you look at the entirety of it, is it's increasing a lot more or magnifying a lot more as you move up your trophic levels, because these are going to be toxins that are not excreted. Metal ions are taken up by producers, or we could say filter feeders. Metal ions are not excreted. Could also refer to them, they are stored in tissues in their cells. And then referring to this as bioaccumulation occurs. If you haven't seen one of my videos before, the slashes, which I may have used already, but the slashes mean you could refer to this, or you could explain something this way. You could also state it another way, but I would definitely say like the metal ions are not excreted, and thus bioaccumulation will occur. Bioaccumulation. And then the next one, we need to talk about it increasing up your trophic levels, because we need to ultimately answer why it's harmful for top predators. So the metal ions increase along the food chain biomagnification occurs. Why else it might be harmful? Metal ions can change hormones, gamete production. Gamete is sperm and egg. They could cause the metal ions. I'll say ions could cause the death of producers or low trophic level organisms, which results in less food availability for the food chain. Woo! All right. That's our question 1, and I'll pick back up on question 2. Question 2, talking about organisms requiring oxygen for aerobic respiration. And a common question, complete the balanced chemical symbol equation for aerobic respiration. And so we need to include the coefficients on this. So we have 6 oxygens. Not me messing it up right away. Oxygen and glucose. And then it will release 6 water molecules and 6 CO2. So 6 for all of it except for glucose. Next part, the fish will obtain oxygen from water by using either pumped ventilation or ram ventilation. All right, pumped ventilation, if asked, which this question doesn't, but if asked, pumped ventilation, we need to talk about pressure and volume twice. They talk about how fish do pumped ventilation. Pumped ventilation, though, requires ATP. It's referred to as an active process because they're constantly contracting their buccal muscles. So they're constantly doing muscle contraction. Ram ventilation, you have continual or constant swimming with their mouth open. And so they're already swimming, so it's like 2 birds, 1 stone. They're already swimming and they're going to just swim with their mouth open. Therefore, they can move and get water that has oxygen dissolved in it to flow over their gills and out their operculum. The rate or the speed of oxygen consumption of a tilapia fish using pumped ventilation was determined, and they placed the fish into a tank of water for 2 hours, and the change of oxygen concentration was recorded. So they measured the starting oxygen concentration, and then they measured it at the end of the 2 hours. The water was enclosed in the tank, so no oxygen could diffuse in from the air. The mass of the tilapia was 0.6 kilograms. And the mass, or the amount of oxygen consumed within the 2 hours, was 525 milligrams. So calculate how fast, or the rate of oxygen consumption per kilogram of fish per hour. And that's very math wordy kind of. So we're going to be looking at a rate just means we're going to have time involved. But oxygen consumption is the milligrams per kilogram of fish per hour. And that is exactly what we need to be dividing. The oxygen divided by the fish mass divided by the time. And they want the answer to 2 significant figures. And it's 2 marks. It doesn't say show your work, but you absolutely need to show your work. All right, so we need to divide the milligram, which is 525. I don't know why I put 5 milligrams of oxygen and excuse me, 0.6 kilograms of the fish. Okay, sorry, my 7-year-old daughter's in first grade, and for some reason she has a friend who is also in her class and has a cell phone and is going to be calling me a million times, which will disturb the screen recording. Love it. All right. Okay, so kind of certain where this got cut off at, but we're going to be dividing the amount of oxygen that's consumed by the size of the fish. And so that first division, 525 divided by 0.6, we're going to get 875 milligrams per kilogram of fish. But if we look at our units, we need to have it milligrams divided by kilograms divided by the hour. And they did this for 2 hours. All right, 875 divided by 2, we're going to get 437.5 milligrams of oxygen per kilogram of fish per hour. But that's not it, because we need to give the answer to 2 significant figures. And right now we have 1, 2, 3, 4. And so we need to knock off some of those. We are going to have to round it. So we can't do 438 and have the 5 round the 7, because now that'll give us 3 significant figures. We need to round it to like its next like 10 whole number. And can't round down. It can't be 430 because it's 437.5 and it's closer to 440 than anything. And that is going to be our answer, is 440, because here is 1 significant figure, here is the second one, and zeros that are leading or zeros that are trailing are not significant. Losers. All right. Scientists investigated how swimming speed will affect the ventilation rate, or how fast they're ventilating, and how fast they're taking up oxygen. So how fast they're swimming will affect the ventilation rate, and how fast they're taking up oxygen. The ventilation rate was measured as the number of times the operculum opened in a minute. And if they're talking operculum, then this is going to be pumped ventilation. Same one was placed into a transparent plastic tube so they could see the operculum moving. Okay, and they do swim against the current. That is their life. The salmon swam against the water current in the tank. The ventilation rate, or how fast they're doing pumped ventilation, and how much, or how fast they're taking in oxygen, were determined at different water current speeds. Okay. A graph. So the results are shown. We have how the water current speed, which is our x, it's our independent variable, is going to affect how fast salmon is going to take in oxygen. And also the ventilation rate, how fast they're doing pumped ventilation, how fast the operculum is moving. All right, before we graph it, I try and, again, make sense of the numbers that I'm looking at. So water current speed, this is increasing. The rate of oxygen consumption. So here is increasing. And then we have a small increase, but nothing like it was before, at the 20, 25, and 30 centimeters per second water speed. And then their ventilation rate, so their operculum movement. here we have the operculum's increasing, and here we have operculum is not doing anything. And so it could mean, did the fish die? Possibly, but then you have over here, you have that they're still taking in oxygen, so they're not dead. What it means is if their operculum isn't opening, they're not doing pumped ventilation anymore. They're not doing pumped ventilation anymore. That means they're doing ram ventilation. And then if I look back at the rate of oxygen consumption, how much oxygen they're taking up, we see initially we have this increase from 112, 116, 17, and 20. And their operculum is moving a lot more. Again, we know that pumped ventilation is an active process, which means they're going to have to be contracting muscles, doing cellular respiration faster so they can have ATP faster so they can have muscle contraction. And that's going to require more oxygen to be taken up. When we don't have pump ventilation happening, so we see these zeros, there's a smaller increase in oxygen consumption because oxygen is not going to be required for ram ventilation as it is in pumped ventilation. So these right here, this is ram ventilation, less O2 required because they're not swimming and contracting their operculum. They're just swimming and they're like, hey, we can actually swim with our mouth open. It's cool, it works. And so we're going to draw a graph of the rate of oxygen consumption and how the ventilation rate changes at different speeds shown. Join your points with ruled straight lines. It's 6 marks. For these marks, typically on an A-level test, it's a 5-mark graph, but what's different with this one is you are given marks for the points plotted for each line. So in the past, it used to be you got to do 2 lines. If you make a mistake in one of those lines, you lose, you suck, you don't get those marks. And now you do have some wiggle room. You could make a mistake in one and not lose it for both, which is kind of nice. Our axes need to be labeled identically. So, and this is where we're going to be using a ruler. We have water current speed in centimeters per second on my X. Okay, and then we're going to be doing a double Y graph. So this one, the rate of oxygen consumption in milligrams per kilogram per hour. And then the other y-axis ventilation rate is the operculum openings per minute. You could also just put per minute, that's also fine. Now we need to have a linear scale. I'll do the x-axis first. So 0, 5, 10, all the way up to 30. And it's going to be too good to be true, but we have 1, 2, 3, 4, 5, 6, 7. Can label each one as 5. Interesting. Oh, it was fun. Always start with your 0. All right, and now we need to put in our other x-axes. So the rule here, it doesn't matter what you count by, it just one needs to be a linear scale, so counting in the same increments throughout for each y-axis side. And the other thing is you need to have a large graph, so it needs to take at least half the space or more. And on the rate of oxygen side, you're going from 105 is your smallest value to 120. So including the value of 105, we need to fit in 16 numbers. If you're thinking, no way, 120 minus 105 is 15, mathematically, yes, you are correct. But we're not looking for the values in between those numbers. We need to also include that 105. And if you don't believe me, you can try it your own way. I don't recommend it. You'll be erasing. Okay, but we are starting with a large number. We're not starting with, we don't need any values between 0 and 104. So we're going to break the graph. It's going to look like this. You could also do these lines. That's also fine. All right, and so I know I need to fit in 16 numbers, but I'm gonna have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. There isn't a calculator function on the iPad. It's brilliant designing. All right, so I need to put in I need to do 16 numbers and split it up 12 ways. Ugh. When I divide 16 by 12, I get a deplorable 1.33. Now you can count by 1.33. Why would you want to? We should round up. You can make this, you can count by 2s. If you're not sure, just try and imagine where those numbers would go and stretch out on here and make sure you're using at least half. You can count by 1.5. It's up to you. I'm going to count by 2. I'll count by 2s. I'll do by 2s. All right. So I'm going to start at 105. This will be 107. 109. 11. So it will take up at least half of the space. Okay, and I'm actually going to graph those values first. That way, if I had this side graphed in, I'm not going to get confused for a moment and look at the wrong numbers. So I'm just going to graph those numbers first. The way you can graph your points is with an X, or you could do a dot with a circle. That's it. Those are your options. I am a fan of the X. That's what I'm going to do. I'm going to use a different color for it, even though I'm just doing it so you guys can see it. Don't ever do this on your graph. This is always in pencil. So my students are like, oh yeah, but I can't see it. But it's not about you. You're not the one grading your own work. So if they want pencil, we're giving them pencil. Okay. So starting at the top, it's 0. The rate of consumption is 112. So 0, 112. I think it'll be helpful to tell myself what I'm counting by, because you need to be pretty perfect. You need to be correct within half of a box mistake, half a box over or half a box under. So for example, if your point was to be right here, you could put it here or put it here and have it be correct still, which pretty much means don't make a mistake. All right, so I'm counting by 2s, and I have 1, 2, 3, 4, 5 boxes in between. So 2 split up 5 ways, each box is going to equal 0.4. All right, so 0 and 112. Here's my 111, 111.4, 111.8, 112.2. So I'm going to be somewhere like right here. Let me erase this. Yes, okay. My next one is 5 and 116, and please speed this up if you'd like. 115.4, 0.8, 16.2, so right here. And then we got 10 is 117, right on it. 15 and 120. So 19.8 is 20.2 right here. At 20, we go down to 105. .107 and then 109. All right. Again, you're using a pencil to do this. So when I make these lines, to split up when you make a key, so you can differentiate between these two different graphed lines you're going to have on here. In the corner, anywhere, you're just going to put key. Now you could do two things. You could say X is going to be the rate of oxygen consumption. And then if you have a dot with a circle, that's going to be the ventilation. Ventilation, there we go, right? Or you could split it up by their lines. So you could say the rate of oxygen consumption is actually my solid line, and the ventilation rate is the dashed line. So you could separate them by the points you're plotting, X or dot with the circle, or by the lines that you're drawing. All right, we connect point to point. Obviously with the ruler, but I have a little cheat. All right, pause if you need to catch up. I'm going to do the next line. So this one, we have zeros and then 42 to 49. We're going to obviously assume zeros down here, but then we have nothing between the numbers 1 through 41. So we're going to break the graph again over here because we really don't need to include those other values. And what the break in the graph really is like an accordion. So like if you took each side and you stretched it out, you would have the numbers 1 through 41 included in it. So we have 42 to 49, so we have 8 values we need to place in. Again, if you're subtracting, you're like, no way, that's 7. You're correct. But we're also needing to include 42. So we do need to put in 8 values. And I'm going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12 to put in. Just for a good time, if I split up the number 8 by 12, I get 0.66 repeating. Nobody wants to count by 0.6s. You can actually just round it to 1. And if you're not sure, then definitely, you know, imagine filling those numbers in and see how far it would go. You need to use at least half the space or more. All right, you could also start at 40, 40, 41, 42, 43, whatever, or you can start at the 42. Doesn't matter. I'll start at the 42. And that's my highest value, is that 49. So I am using half the space or more. 50, 51, 52, 53. You didn't have to number every 5 boxes either. You could have done every 10 if you wanted. All right, similarly, I really don't need to count and see like what each box is worth because I don't have any values in between. They're all their whole number. But if you did, I'm going from 42 to 43, for example, so that's a jump of 1, and then I have 5 boxes splitting it up. I would do 1 split up 5 ways. So 1 divided by 5, you get 0.2. But you won't have to do that because these numbers are all actually on those lines. Cool, great. I was looking for a different color. I'll do red. I'll live with it. Don't use color on your test. Okay. I'm actually going to separate my points by the line thickness. Like, not separate my points. I'm going to separate my different lines that I've graphed by line thickness. So the one we just did, the rate of oxygen consumption, I'm going to keep that solid. And then for the ventilation rate, I am going to do X's, but I'm going to do it as a dash line. Actually, I'll do it as purple as well so you can see the differences. All right, so we have 0 and then 42. So 0 down here, 42 over here. Just confused myself for a second. I'm back. Five and 45, so 5 up to the 45 right here. 10 and 47, 47. Read it across at the 10. 15 and 49. Zeros for 20. For 25 and for 30. All right, now I'm going to connect them. So they're following the same trend so far, both increasing as the water speed increases. And then we have a big fall. Okay. And you want to make sure that you have a key that suits what you're doing. There it is. Never a title or anything like that. And where we have this big drop right here, that is going to be where the salmon switches from pumped ventilation to ram ventilation. But they both do follow the same trend. Our question II. Describe, so that means what is the effect of increasing the water speed on how fast they're taking in oxygen and the ventilation rate of the salmon, how fast that they're opening and closing their operculum. And for this, they both are doing the same things. Both oxygen consumption and ventilation rate increase. And then decrease. Now, if you didn't say that in a sentence where they both, and you did separate it, you just said the rate of oxygen increases and then decreases, the ventilation rate increases and decreases, you could still get this mark. And there's an area where they both start to follow this trend, and it's at the water current speed of 15 centimeters per second. That's going to bother me. No, I'm fine. All right, the salmon swims at the same speed as the water current speed. So we can assume that as the water current is increasing, the salmon is going to start to swim faster. Use the information in the table and your graph, which is the same, it is the same data. You're just have a visual of it. Suggest reasons or suggest explanations for the effect of water current speed. So what happens, give reasons for when you increase the speed of the water on how fast they're taking in oxygen and how fast their operculum is moving, so the ventilation rate. You should refer to ram ventilation and pumped ventilation in your answer. So this is not the only way you can answer this question, but this is how I'm going to go about it. First, I'm going to compare both methods essentially, because that will kind of set the stage for why we see the oxygen consumption go down whenever they switch to ram ventilation. It'll also give reasons and kind of set the stage for why we see the operculum or the ventilation rate go down. So this first thing I'm going to mention is kind of what they are and yeah, just what the difference is for them. Pump ventilation uses the opening and closing of the mouth. Or you could talk about pump ventilation uses buccal, or you could say buccal. I always say buccal. Muscles to pump or force water. Over the gills. Okay, and we need to talk about ram ventilation as well. So we have to compare them both to get this mark. Ram ventilation uses something along these lines, like an open mouth, parentheses, just to add context, and constant swimming. Constant fast swimming, I would include that. To force water over the gills. So we need to talk about water current speed, and we can talk about the need for oxygen, and then water current speed and operculum movement. They need oxygen to do respiration, but that's because they're constantly contracting their muscles. As the water current speed increases, there's more muscle contraction. And we're going to run out of space, obviously, in the lines, but you can use the space below here to continue answering. Okay, but this is up to 15 centimeters per second of like the water speed. Pumped ventilation, increase the supply of oxygen to the gills. All right, we did in the very first statement compare buccal muscles or muscle contraction for pumped ventilation. And then again, we're explaining, anytime we're talking about oxygen, always talk about respiration. Pump ventilation requires more energy. NRG is just my shorthand for energy. We're not going to write that on a test either. Again, in parentheses, just for context, due to increase muscle contraction of their buccal muscles. I'll just add here too, which will require more oxygen. If you need to do more muscle contraction, you're going to need more oxygen. We essentially already said that. Okay, and then we need to talk about continuing the speed. What's happening as the speed continues? As the water current speed increases past 15 centimeters per second, the salmon switches to ram ventilation. Why would it switch? Like, why didn't it do it the whole time? You could say something like the slower water current. So anything less than, like anything less than 16, right? We don't have data for 16, we have data for 20. But at 15 is, anything higher than 15 is when they switch. The slower water current isn't fast enough. To absorb oxygen. For ram ventilation. Slower water current isn't fast enough for ram ventilation. Okay. Now, these sentences don't have to be just like how I wrote them. You could use alternative wording to describe the same thing. And that is it for question number 2. All right, for number 3, this one is going to cover salmon aquaculture. All right, so it reads salmon are grown commercially around coastal areas. They're using extensive aquaculture on salmon farms. So outline the process of salmon aquaculture in extensive systems. We're going to have to make sure that for extensive, we talk about natural water flow and in general, consider the general process for this. So we need to have like a starting population. We need to consider what's going to happen with the juveniles, or at least mention the juveniles. I suppose the larvae stage first. And then your adults. And it starts over again. But we're going to need to talk about just in general their care. So what will their food sources be like? How will they get water? Flow for oxygen, for waste removal. All right. So with our starting population, that's our brooding salmon. Most aquaculture ventures will keep brooding salmon or salmon that are able to reproduce. All right, they're used to produce. You could say gametes, or you could say sperm and egg. And then those eggs are fertilized, and you will have fertilized eggs, and then they'll be in their larvae phase, so 11s, and even like small fries, those very small sizes, just to encourage their best growth potential and minimize predation and competition. They're going to be kept in hatcheries, or they're kept in like smaller tanks. Oops, I'll spell that right. I'm just going to put for context for their specific ages, or you could say that they're kept in hatcheries. I know salmon, like their fertilized eggs specifically, are kept in like cooler water in those really like long pans. They almost look like cookie sheets. We're not going to say that. We're not going to say cookie sheets. This is just outline. All right. After that, juveniles. Are transferred to outdoor cages. You could also refer to them being in like ponds. Does not have to go necessarily in this order, but this works. The fish are, I'm going to write separated by their age and size. because their sizes will require them to have different food quantities and different types of food. And we definitely are going to separate them by their size, otherwise there'll be competition with adults or the age that's larger than them, and they will cannibalize each other. With that being said, food is added specific to their age or specific to their size. So the food is specified. We are going to want to reduce any sort of disease potential. So pesticides for parasites, vaccines, antibiotics. Are used to prevent disease. Another threat to them could be predation, not maybe amongst each other if we're separating their ages right, but from outside sources. So that could be netting within the water or netting on the top so that birds can't eat them. Nets are used to prevent predation. And we still need to take care of this extensive term. So I'm going to mention that the outdoor, the natural water flow is going to supply oxygen for respiration, remove waste, can also help supply food. And since this is a commercial venture, if this isn't a fully extensive system, then they're going to want to supply their own food as well, but they're also going to take help from the environment, of course. Natural water currents brings oxygen for respiration. Always link oxygen with respiration and respiration with the need for oxygen. Likewise, we would link sunlight with photosynthesis. And if we talk about photosynthesis, we're going to talk about sunlight or light energy. Waste removal, and also supplies food. All right, one of the problems are the potential for escaping. It's only one mark. Scientists investigated the escape of salmon from fish farms to the wild. Farm salmon are fed food pellets that contain dye. The dye accumulates in the body tissues of salmon. Salmon are caught by rod and line. How sustainable? From rivers and areas of the sea close to a salmon farm. Salmon that had escaped from the salmon farm were identified by the presence, identified by the presence of the dye in their body tissues. And this graph shows the portion or the percentage of female salmon caught that had escaped from the salmon farm from 2000 to 2005. So the percentage or the portion of salmon that were caught that were actually from the farm. And they knew this because of the dye. And just looking real quick, just to identify a trend, we can see over time, there starts to be more of an escape. But that's something to consider too. You don't know that for sure. The bars show that they're increasing over time, but does that mean that they're escaping more? Like, are we just not learning our lesson and getting worse with trapping them? Or you may have some that escape that just have still persisted in the environment and haven't died. It's possible that they're not necessarily escaping more, but we might have more fish farms for them to have the ability to escape. Things to consider. So in 2005, a total of 53 salmon were caught, and they're not specifying were they wild or were they escaping from a farm. Just 53 were caught. Calculate the number of female salmon caught that had escaped from the salmon farm. All right, and this is in 2005. So we're going to be looking at this bar. When you consider what we're counting by, because we're going to have to read up here. Okay, so I think this one's obvious. They're going to be counting by twos, but in the case that it wasn't so obvious, you have 1, 2, 3, 4, 5 lines, but you're going from 0 to 10. So you have a jump up of 10, and then you're splitting it up 5 ways. So each box will be equal to 2. And over here, all right, we're at 80. Right here, 82. 82%. So there's 53 female salmon that were caught, and we know that of those that are caught, 82% had escaped, and we know this from here. The percentage of female that caught or escapees. All right, though it doesn't ask you to show your work, always show your work. 82%, just to write it out for you, escaped. From the 53 that were the total catch. We're going to do 53 times 0.82. All right, and when you do that, you get 43.46. And you can't really have a 0.46 of a salmon, but that's not quite 0.5, so you can round down on that and call it 43. It's not 43%, it's 43 salmon. And just what we discussed. So why would the percentage of female salmon caught escape increase from 2000 to 2005? So there's potentially more salmon farms over time. Or kind of along the same lines, within those farms, there's more adults kept. As brood stock. And we only need 2 marks here. The salmon don't die just because they escape, you know, so they're going to live for more than a year, at least. And that can cause us to increase. So maybe, you know, a handful escaped 2002, but then those are still surviving in 2003, and then 2003 has some of their own escape. And so, I mean, you could also say this as, you know, their escape population can build up because it's not like they die as soon as they escape either. Potentially one of the more easier answers is that maybe just more escape every year. And then we can like give a reason why. From damage to cages, bad weather. Bad weather ruining the cage. If you're a student in my class, then maybe you recall in the aquaculture notes, there's, and actually, I think everybody has access to this on my YouTube page, the aquaculture notes. I think those are published. But in Seattle or Washington area, there was a salmon farm and a storm damaged their external setup. And then I think there was like 300,000, if I remember correctly. It was a large number that were able to escape into the wild. I'll try and squeeze in some more. So what if it's not necessarily that there's more escaping? Maybe the ratio of wild to farmed fish is changing. Maybe that there's less wild fish available. So numerically, it might seem like there's just more farm fish escaping, but in reality, the ones that they're catching are going to be farm because the wild are being overfished. Let's see, wild salmon are overfished. I'll squeeze in by it. It's another mark. So, it doesn't have to be overfished. I'll give another optional one too, but this is just kind of regarding to why there's maybe less salmon, wild salmon in the population. Less wild salmon in population. And one of those reasons could be that the wild salmon are overfished, or potentially maybe there's some toxins or pollution or eutrophication maybe in the Red River beds, so their eggs don't develop. All right, the biological drawing. So question 4 about mangrove forests. Obviously are an area of environmental importance. They are areas of great species diversity and habitat diversity. They also are nursery beds. Replanting mangroves has been suggested as a method to increase biodiversity of the areas they grow in. Also, replanting mangroves helps with shoreline restoration because they slow down wave and water current energy and help to increase sedimentation. So it rebuilds up habitats. It also is an habitat restoration, also like shoreline erosion. It's also being used a lot, like right in Dubai, they have the biggest plan right now to have the largest mangrove forest. It's pretty interesting, but they also store much more carbon than a traditional rainforest. So something that I recommend is you want to stay in proportion with things. So right away, I would try and measure like kind of the radius and how wide it is. So it's just about like 3 centimeters. You can call it that. And so I would say like at least double it because you want to make it, it says large. So I'm trying like at least double it. And use my pencil here. Is that what I had? 3 centimeters? What the heck? Yeah. Okay. So I'm going to go from like here to over here. Okay. Get this out of the way. No, I'm going to make a cheat, obviously. All right. There's nothing to say that you couldn't bring a protractor, but If it's drawn in a circle, I like to also duplicate the circle. And something else that I try to do is like, sometimes I'll draw over it just to get the feel for what I am drawing. And things to point out, we can see that there is this double layer here. We'll want to include that, but no shading, nothing like that. I think the hardest part is proportion, and so I try and at least start with double whatever size I have. So we don't have a ton of like area like right here exposed. So kind of do something like this, and please speed me up. There's a double layer. I typically would have saved that for last, but here we are. Okay. All right. There'll be a lot of erasing, which is why we want to use our Ticonderoga. The next things I'm going to draw in is like this right here.
Attention, students and staff, pardon the brief interruption. Just a reminder, when the bell rings, all students will report to fifth period.
Got that? All students report to fifth period when the bell rings.
To fifth period prior to going to lunch. I will come back on the announcements and release for a lunch, but all students will report to fifth period. Thank you so much and have a great day.
Okay. And we want non-broken lines. I think if I erase, I'll erase the whole thing. I don't want to. All right. And sharp pencils. Sharp, 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 sharp pencils. Sharp pencils. You can bring a little handheld sharpener in with you. Obviously one that doesn't make noise. You can bring that in with you. Just has to be like clear. And then we need to put in our cells. So we have a cell right here. It's small. Visually, we can see that like this one and this one are the largest. And then we got little baby here. She's skinny. Also here, here. Sometimes these are like the size proportions that they look for. And this one is pretty large. It comes almost to the side, and then we got a little edge right there. And there are some nuclei in them. I'm gonna do my big ones first. And this one kind of hits right where the center is. This one hits there. And there's a gap at the top. Okay, actually, it's more like this, doesn't come down that far. And actually comes a little bit further over. And I want to show you too, like this right here, ugh, you're going to want to get rid of that. All these unneeded extra lines. And you notice it's even wider than that. All right, then we have one right here. Oh, stop it. Kind of comes in a little bit. I'm not pleased with any of that. Ugh. It'll be fine. It'll be fine. And this one kind of turns. Here's a large one. No, don't assume a circle. Thank you. And lift that up so that it doesn't draw a circle. Do I love it? Of course I don't. Am I going to be okay with it? I have to be. I'm not submitting this to Cambridge. that's not going to work. Okay, like this should be fully connected. This should be fully connected. I'm going to erase some of this so I can really see where the nuclei are. Cool. All right. Here, here, a little one down here, here, and here. Okay, I'm going to put cell walls on all of this. This isn't something you want to use use a ruler for to get straight lines. And even like if you see like this space right here is not that as big as I have it, I probably would go in and make it a little bit longer, but I do have a class waiting for me outside the door. Like elongate it here and elongate it here a little bit. But my class. Make it a little bit thinner. This is a silly song. Can I do it? No way. Okay, and then finish the cell walls on these guys. All right, I'm gonna have to live with it. I don't hate it. I don't hate it. Okay, my students came and went. They'll be back from lunch soon. But just looking here, like just to reiterate, I would, honestly, I think the easiest thing to fill like this gap right here is just not the correct proportion. I would actually like take some of these off and then move them over. And they're so particular, which is why we definitely want to have a wonderful eraser and not draw extremely heavy the first time. You know, and like even, I'll erase this, but like some of these lines right here, I don't think I would, that one, I don't think I would be submitting that as my official like sample. But we want to have every line touching, so nothing wispy like this. We don't want any sketch lines either, nothing sketchy. It's not respected. And like this right here, I would definitely try and fill that in. But just for the sake of practice, I'm not horrified at it. Okay. I'm like, I need to move on with my life, but this is longer than it is wide. The way I had it drawn, it's pretty circular. So maybe do some like elongating. That might be better. All right, so Those are things I look at, but it's never perfect the first time. Let's do some Spearman's rank. So they wanted to see if a mangrove, oh, did I say Spearman's rank? Yeah, no, it's exactly what we're doing. All right. The size of mangrove forest affects biodiversity. All right, so this is to see if two things are correlated together, but we also know that correlation does not equal causation. The student collected data from different size mangrove forests. The student then carried out Spearman's rank correlation coefficient test to test the null hypothesis. The null hypothesis was that there was no correlation between the area of mangrove forest and the Simpson's index of diversity of organisms. So interesting, they're using both tests here. Simpson's index of biodiversity, it tests to see the biodiversity of an area. So that ranges for Simpson's, it always ranges from 0 to 1, 1 being very, very diverse. Biodiverse would be like a coral reef or a well-developed mangrove forest. Could also be like a rocky shore, maybe like a subtropical rocky shore. And 0 is like your sandy shore, hydrothermal vent, like no biodiversity. It'd be a very low number. Spearman's rank is carried out to see if two non-normally distributed data sets correlate together. So by non-normally, it's like you don't see things like in a numerical order. So you couldn't say, well, as this value increases, this value increases. They're not set up like that. The way that the numbers are distributed are based on the habitat that they took these samples at, not by their numerical like ranking, smallest to greatest, nothing like that. Okay, so what they're ranking then is the rank of the forest area and then what they ranked in Simpson's index of biodiversity. So they're ranking it, and you can tell by how they start it. Their first rank, number 1, is going to be their largest area of mangrove. So that's the 0.11 right here. So they have 1, and then over here is 2, 3, 4. So this would be 5, and this one could be 6, but we can't have any, like, you can't really say which one's 5, which one's 6. So in that situation when there's a tie, you're going to take both of the next rankings. So the next ranking would have been 5 and then 6, and then do their average. So 5 plus 6 divided by 2. You're going to have 11 divided by 2, so 5.5. So we see 5.5 here and 5.5 there. So that's your 5 and your 6. And that's for 0.04. My next ones then is 0.02. But I have 0.02 and 0.02 here. Ooh, how fun. This is the first time we've seen this, where there's a 3-way tie. The method to break this tie is going to be the same. So our next ranks we need to look at, we have 5, 6. So our next ones would have been 7. Here we have 8, and then 9, and 10's already done. But we can't really say, okay, you get to be 7, you get to be 8, you get to be 9. Because those numbers are going to complain. They're going to whine about it. They're never going to forget the day that you wronged them. I wanted to be 7. No, you can be 8. They're like children. So we're going to do the same thing. We're going to add these up and then divide by 3. 7 plus 8 plus 9 is 24. And then we divide by 3. What do you know? We get 8. So this is going to be 8, and this is going to be 8, right? Because that's the average of 7 plus 8 plus 9, which are the following rankings that will go with 0.02, 0.02, 0.02. And then we're going to take their difference. So we're going to subtract R1 from R2. They've already done some for you. So we need to be subtracting 8 from 10. And then 8 from 8. And then we're going to square those differences. All right, so 8 minus 10 is -2. And if you're like, well, can it be positive 2? It literally won't matter because we're squaring them. Negative 2 squared, or even 2 squared, is 4. 8 minus 8, 0. 0 squared is 0. And now we're ready to find the summation, or the put-togethers, the adding, the sum of all of your d squared. So 1 plus 0.25 plus 4 plus 0 plus 0 plus 1, 1, 1, oh sorry, 1, 1, 0, and then another 1, and then 0.25. If you add them all up, you're going to get 8.5. All right, and now we're ready to do our actual Spearman's rank formula. They'll always give you the formula, you just need to know how to apply it. All right, we need to do 1 minus 6 times this, the summation of d squared, divided by n cubed. And they say the number of paired items. This is, again, this is like your sample. And they looked at 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. They looked at 10 pairs. Their pairs were the area of the mangrove forest and the Simpson's index there. So it's going to be 10 cubed minus 10. All right. Again, we have 1 minus 6 times 8.5, so why we need a calculator is 51. 10 cubed is 1,000 minus 10 is 990. When we divide that, and move it next to it, we're going to have 1 minus 51 divided by 990 is 0.51515. Don't round early. Don't round early. Round at the very end. When we subtract that, we get 0.94848, and they say give your answer to 3 decimal places. Sometimes they'll tell you the amount of significant digits, but they want 3 decimal places. And so the answer will then be 0.94. If I'm looking at the 8, this 4 is not going to make it a 9 or round it up or anything. So there it is. And now we're ready to calculate or compare our calculated value to the critical value in the table. All right, so we just need to have our n, which is the number of pairs, and that is 10. And so the way that we read this, and we always look, if they give you multiple columns, we're always going to look for a p-value of 0.05 or 5%, which means that there is a 5% probability that these correlations are due to just chance and it's random. They really don't correlate together, or they're not related to each other. Or there's a 95% confidence that they do correlate together and that they are related. All right, so looking at the n column, we were looking at 10. So this 0.648 is the critical value we need to we need to compare with our calculated value of 0.948. So the calculated value is greater than the critical value. And if our calculated value is greater than the critical value, that means that we are going to reject the null hypothesis. And looking back, the null hypothesis said that there was no correlation between the area of mangrove forest and Simpson's index of diversity. Therefore, that saying that like area of a mangrove forest does correlate to how biodiverse the area is. The null hypothesis is rejected. Now, if our calculated value was smaller than the critical value, then we would accept the null hypothesis. You could put the calculated value is greater than the critical value from the table. You get a mark for being able to read the table. So saying what the critical value is. And if you said this in like one statement, the calculated value of 0.948 is greater than the critical value of 0.648, you would get 2 marks for that statement because you're referencing the actual critical value. You get a mark for that, I imagine, because, you know, you could always just be like, I don't know what I'm reading on this chart. I'm just going to wing it and guess and say something. So you do get a mark for proving that you know how to read it. All right, and so therefore, there is a strong, or you could say significant, and the 0.948 is a strong positive correlation. Because it is very close to 1. BTWN, between. Mangrove area and species diversity. And then we can also throw in there, though you don't have to because this is 3 marks, but there's a 5% probability that the correlation is due to chance. All right, but again, correlation does not mean causation. You could include that too. And number 4, suggest why, so give reasons. The size of the area of a mangrove forest will affect Simpson's index of biodiversity. So essentially, how come if a mangrove is biodiverse, why does that mean it's going to have a higher biodiversity? Did I say that? Why, if the size of an area of a mangrove, did I say that right, is high, why would it affect Simpson's index, or why would there be a high diversity? So a high diversity from high mangrove forest area means there's more producers, or there's higher productivity. And the only reason there's species there anyway is because there's food. Like the food chain always follows. If there's an area where there's not going to be like stable substrate, like a sandy shore, that allows for plants to grow, then you're not going to have a very biodiverse area. But if you have a substrate that does allow for plant growth, then it can allow the rest of the food chain to follow. Okay, so just again mentioning about the producers is going to be 1 mark. It will also mean that there will, so this causes more energy in the ecosystem. Which causes more trophic levels. And you don't have to write it with bullets, but these are just where the marking points are at, just mentioning these. There's going to be more niches or roles for organisms to occupy. More habitats or nesting areas. Shelter from predators. Food sources. Shelter from storms and really strong water currents. All right, this one is lovely. And then we'll go into some photosynthesis and then experimental design, and that'll be it. This is the last one for question 4. So a further study in coastal area of Indonesia, local people were asked to complete a questionnaire about how willing they were to take part in replanting mangroves. The result is below. All right, so let's pick out some high data from this. Are you willing to participate in a mangrove replanting area? So willing to participate. We have 12 disagree, strongly, disagree, agree, and then strongly agree. We have much more that strongly agree. Do you currently volunteer to help mangrove replanting? 38 strongly don't, 42 don't. They're not sure if they currently volunteer. 7 and 5 are agree. So we have more that are not currently volunteering. Should you have appropriate pay if you participate? 9, 18, 3, 44, and 26. But we have a lot that many more say that they do want pay. Do you have the skills needed for mangrove replanting? Because you do need skills to be able to do this. And majority of them do not. 5 are like, yeah, I don't know if I know this. The unsure is funny. All right, 3 marks. Use the results. Discuss how government could successfully engage local communities in replanting the mangrove forest. So how could they do this? All right, so we could start off with just saying the importance of getting locals involved. They are main stakeholders in their community. And they'll encourage the rest of the community to do it. Potentially, it's better than the government telling you that you have to. See, local. I'll finish that off. I'm sorry. Compared to government order to do it, or legislation saying that everybody must do it. But if you get the community involved and have them benefit and see the benefits from it, seeing the benefits from it ecologically might not be immediate, but the money would be. They need to be paid for their time. Education and training would be needed. It's because there's many without the skills needed. That 35 and 28%. And you're more than welcome. I would encourage my students to do it, to actually use the data to support it. It's not necessary, but anytime there's data in a table and you got to talk about it, talk about it. 35 and 28, so 50, 63. It's 63% without skills. And after you train up one group, you might not have to continually train up multiple more groups because the locals can train each other. Or encourage more social engagement with the project or with replanting. There is not many people already involved. What is that? 70, 80% not involved. Many are willing to participate. %, coming from right here. All right. And then we'll start with 5. All right, the rate of photosynthesis of producer organisms can be affected by many abiotic and biotic factors. The light-dependent stage of photo occurs in the lamellae of chloroplast. Okay, that makes sense. All right, I was going to say the thylakoids of the thylakoid membranes. Anyway, I'm not here to argue. Give the two products of the light-dependent stage that are used in the light-independent stage. It happens in the stroma. It's also called the Calvin cycle. Common question that we've seen now for a couple years. It's going to be, it doesn't have to go in this order, ATP and then NADPH, or you could say NADP reduced, because it's carrying that electron, which is reducing it. My students just typically know NADPH because it's carrying that hydrogen over to the stroma for the light independent reaction. All right, and then we're looking at a graph that's showing the absorption of different pigments extracted from 2 different species of algae, species A and species B. And just maybe to make it visually easier, I'm gonna throw some color on it. So species A is this gray, the solid gray line. Oh gosh, forget it. Forget it, I'm not gonna do it. Species B is your dotted line. And it says use the figure to explain, so give reasons for which one of the 2 species of algae is adapted to live in deeper waters. So deeper waters, we're going to be wanting to consider which one can absorb the blue and the green wavelengths, because we know that those are the colors that are going to penetrate deeper, where your longer wavelengths with lower energy, your reds, your oranges, your yellows, those are pretty much going to exist at the surface. And chlorophyll is the primary pigment in photosynthesis, and every species that does photosynthesis is going to have chlorophyll, But if they have accessory pigments, then they are going to be adapted to living deeper, so they are able to use and absorb the light that's available there. All right, if we're looking here in your green spectrum, species B just has chlorophyll. You can tell because species B is only absorbing in like this red, orange, yellow area. And then you don't have a lot of absorption within your green, and then they're also absorbing a lot in the blue area. And that's just for the dotted line species A. Nope, species B, I'm sorry. Species A is the gray solid line, and we can see that they are able to absorb same as species B, but they also can take in the green wavelengths. And again, green and blue have more energy, so they're going to penetrate deeper in the water. Any species is able to absorb the blue wavelength that is in deeper waters, but if you have an accessory pigment, you can absorb blue and green, and then you're better than everybody. All right, so it's going to be species A. We'll say has accessory pigments, or you can give the name of a pigment. You can. You could also just say accessory. Parentheses, in addition. I'll spell it right, I suppose. Goodness gracious, great balls of fire, my gosh. In addition to chlorophyll A. If you wanted to call a certain accessory pigment that they have, you want to look at where species A isn't really absorbing much, and that's going to be where that's the color that they're going to reflect. And that's going to be this area right here. Right here. That's where they're absorbing the least. So they're going to show some reds, yellows, oranges. So maybe it's like a xanthophyll. Maybe they have carotenoids or physoerythrin. All right. Species A can absorb more green light or light that's within like this 5, 10 nanometers over to like 640. It's this space right here, kind of like exactly where species B isn't. And now we need to just link this relationship to the depth of light in the ocean. Green and blue light is available or penetrates deeper in deeper waters. Or you can say the reverse argument. Red light or your longer wavelengths. It's available in the surface or at the surface. All right, and now we have our experimental design. How long do I have? 23. I have 6 minutes. We'll see what we can do. All right, no matter the experimental design situation you're thrown in, they're all going to follow this pattern, and it's worth doing it well because they're worth mucho points, either 11 or 12. But atrazine is a weed killer. It's used in agriculture in some parts of the world. Atrazine is a toxic chemical that will inhibit or stop the light-dependent stage of photosynthesis. That's no good. There have been concerns that atrazine can cause pollution due to it being washed into the rivers and coastal waters. Plan a laboratory investigation you could do to investigate the effect of. That is going to be your independent variable. Changing the concentration of a weed killer. That's your independent variable. On the, so how it affects essentially. On the rate of photosynthesis of an aquatic plant. Rate of photosynthesis, that is, or how fast they're photosynthesizing, and we need to know ways that we can actually measure that. The rate of photosynthesis is going to be your dependent variable. That's what you're measuring. You are provided with a 1% stock solution of atrazine. So you're starting with a concentration no higher than 1%. That means that any concentration examples you give cannot be more than 1%. That is impossible. So this can't be greater than 1%. All right, and you'll see what I mean by that in just a moment. Your plan should have a clear statement of the hypothesis. Identify the independent, dependent, and standardized variables. In America, sometimes we refer to standardized variables as the control. I've actually seen it referenced both ways for Cambridge. I feel like they're trying to say standardized for things that are kept the same, but I have seen it in the mark scheme called the controls, which is a little contradictory. True control variables are going to be variables that you don't do, or like a trial that you don't do anything to. You just allow it to be the normal. Sometimes you can do that. Maybe if you wanted to see the effect of changing salinity, and one of the salinities is the average ocean salinity of 35 parts per thousand, and that would be like your control to compare the results to see if there was any kind of change. Okay, so standardized variables are things we're going to keep standard, keep the same. Sometimes they give you a prompt like this where it says, say the independent, say the dependent, say the standardized variables, or they just say list the variables or include the variables. Details of the method, so another person can follow it. Describe how you would analyze your results, and there's a few blanketed statements we can say for all of our experiments, honestly. Within those results, we can also include a data table. We'll need a ruler for that. Also, like what a graph would look like or how you would label a graph. Statistics. Another thing we can mention, be safe and be ethical. So we always want to mention something about safety, mention things about ethics. If there is not an organism used in this situation, there will be because it's a plant. But if you do not have an organism used at all, maybe you're just doing something simple, I don't know, temperature and salinity, great. Or like dissolution. Then you can just give a statement and say there's no ethical protocols or no ethical considerations because live organisms are not involved. And you can still get a mark for saying that because at least you're considering the ethics. All right, so we're going to start with the hypothesis. This question is 11 marks. In order to get all the 11, you have to have the hypothesis. So the hypothesis is absolutely one of them. Okay. And it doesn't have to be like an if-then statement that's very America. It doesn't have to be like that. And ideally, they want to see, again, what is the effect of changing the concentration of atrazine on the rate of photosynthesis? That is what our hypothesis needs to pretty much include. So I'm going to say as the concentration of atrazine increases, the rate of photosynthesis decreases. All right, and then we need to go into our variables. Okay, so this is what we're changing. This is going to be the concentration of the atrazine. And that is a mark. Five examples, 5 realistic examples. If there was one about the effect of decreasing the pH or acidification on oysters settling to a substrate, and some students gave examples of a pH that's higher than the average of the ocean. So a pH that wasn't even acidic, you would consider it to be alkaline. So that's where they need to be realistic. If it was temperature, we're not going to take it to 0, that's freezing. We're not going to take it to 100, that's boiling. So we want to be realistic. So we're going to give 5 examples. Keep in mind, we cannot have anything that is greater than 1%. So we can say like a 0.2%, 0.4%, 0.6%, 0.8%, and then your 1%. If you can, if it makes sense, and in this situation it does, you can list what the control would be. 0% or no atrazine. And a control is something you're comparing the results to. Things that are standard are things that are not changing. Dependent. Okay, and again, you can do this for all of your experimental design, giving an example. That's just another mark. Dependent variable is going to be the rate of photosynthesis. Or how fast plants are photosynthesizing in a certain amount of time. How can you do this? We can count bubbles in a set time, or we can measure the volume of oxygen produced in a set time. Maybe you're doing this like within like 5 minutes or something. And now we can go into standardized variables. And as I mentioned before, standardized variables are the ones that are not going to be changing. And the only thing we're changing is the concentration of atrazine. So everything else involved in the experiment, and consider that we're going to mix the atrazine with water, especially to dilute it. Everything else is going to be the same. You only want to be changing one variable. For the standardized variables for this specific question, you could only get a max of 3 marks, which means these are some of your easiest marks, honestly. You can go for 3. And I typically have my students do at least 3 or 4. Okay, so we'll want the same volume. And in this mark, they also included the salinity of water. We don't want to put amount of water. We're better than that. We're not going to say amount. It needs to be volume. The same pH. We want a constant temperature. And we can implement having a constant temperature by using like a water bath. And that's not a mark here, but it will be in the methods. Within the plant, we want that to be the same. So the same size, species, mass of plant. It's a plant, and so we want to do rate of photosynthesis. We want to make sure that the light intensity is going to be the same. Or wavelength or distance from the light source. Hi. Almost done. The same volume of sodium hydrogen carbonate, which is something we can give to each plant to make sure, or each trial to make sure that they have the same concentration of carbon dioxide. You can put carbon dioxide concentration, because that is a reactant in photosynthesis. All right, and now we're on to methods. Things that we don't need to include are things that are obvious. So clean up any spills, clean down area. That's obvious. But we need to create these different concentrations. So specifically for this one, you could talk about how to create these different solutions and how to dilute them. All right, and so I said could use 20 milliliters of the stock solution, which is the 1%. They didn't say how much you were given. They just said this is the concentration of atrazine, 1%. So 20 milliliters of the stock solution. And dissolve it within 100 milliliters of water. Oops. .2% solution. Likewise, I'll squeeze it down here. $40 milliliters of the atrazine in 100 milliliters of water. We'll create the 0.4%. The 0.8%. 80 milliliters of atrazine and 100 milliliters of water. And I would write this out a bit more. It is, thank you. Mm-hmm. And then, sorry, my custodian isn't here. 0.6%, 60 milliliters of atrazine, and 100 milliliters of water. All right. All right. We could talk about how we're going to place the plant inside. So placing the cut end, or like the shoot, if you know the word, of plant, cut end up in a beaker or a test tube, also referred to as a boiling tube. So it would look something like this. There's the cut end. And that way we can watch to see like when any bubbles come out, count them, or you can also trap them and collect the oxygen created. Very specific detail. I'll include it because it's in the mark scheme, but I wouldn't necessarily think about including this. This is very specific, but use a paper clip. To weigh the plant down, because if they do kind of get saturated with oxygen inside their tissues, then they will start to float. So the paper clip being like right here, just like attached to the plant. Maybe one of the leaves holding it down. All right. Place the test tube or boiling tube, or if it's a beaker, in something larger, in a water bath. So maintain the temperature and allow that to actually be something that is standardized. And so that then would look like this. So placed in a larger beaker. All right, right? Because it's easier to change the temperature of a smaller volume of water, so the test tube, but it's not as easy to change the, to change the test tube, you would also need to change the temperature of the water that's surrounding it. So that's why we use a water bath. Almost done. If you don't want the pH to change, you could say use a pH buffer solution. It just helps prevent the pH from being changed. Nope, not to control, but to maintain. All right, and then how are we going to analyze the results? All right, we are always going to say we're going to repeat the setup, repeat the methods, repeat the experiment 5 times, and We need to include this too. Like, what's the point of doing it so many times? It's so you can get a good average, and we're going to call that the mean. Calculate the mean. Okay, so for example, we're going to have, and I'm going to erase this, but it's going to look like this. Beautiful test tubes. Okay. So we would have this for like the 0.1%, 0.1%, 0.1%, 0.1%, 0.1%. And then we would do the same. Hold on now. Oh no, 0.1 is our highest concentration that we have. No, it's not. No, it's not. You have a 1%. Okay, 0.4. All I'd say is we would have, like, I always tell my students do 5 different independent variables. And we said 0.2, 0.4, 0.6, 0.8, and the 1. So 5 different variables will keep you safe. And then we're going to repeat it at least 5 times. That'll also keep you safe. I think sometimes 3 is what they say, like at least do 3, but you can't go wrong with going over that and doing 5. So you would have this set up essentially for every single concentration. Beautiful test tubes, but 0.2%, 0.2, 0.2, 0.2, 0.2. You would do it for all of them. So you would have a total of 25 test tubes. So with the analysis of the results, we need to talk about how we're going to get the actual rate. And if you hadn't mentioned it in the dependent variable, like we're going to calculate the rate of photosynthesis, and you were going to say how you were going to do it, then this would be the time to do it. Calculate. Dividing amount of bubbles or the concentration of oxygen, like the volume of oxygen that you collected, by the time. All right, and you could actually state this one first. It really doesn't matter. It doesn't matter the order you say it. Talking about calculating the rates and then finding the means. All right, and then we're going to apply a statistical test. This one is something that you can say no matter what. Same with this first mark. Repeat the methods 5 times and calculate means no matter the experiment. That is a common statement you should say. And we're going to apply a statistical test like standard deviation, or Spearman's rank, maybe to see if the concentration is actually affecting the amount of bubbles, the rate of oxygen. Giving an example of what the results table would look like. Now, this mark is tedious to get, so if you're short on time, I would not try and focus on it so much. But essentially, when I try and do this, this is usually what I'm thinking. I'm going to go down here to show you. Okay, so we're going to have these and the 0%. So the control. So we're going to have the 0, which is the control, 0.1%. I keep doing it again, I'm sorry. 1.0%, 0.8%, 0.6%, 0.4%, and then the 0.2%. Okay, so we have these essentially 6 because of the control. And then we're going to have trial 1, trial 2, trial 3, trial 4, trial 5. And this is not anything that's going to be a finished product at all. Just going to show you like what we're looking to try and fit in. And sometimes I'll do this too to be like, well, what would this data look like? What am I trying to collect? Okay. We don't have data to put in, so don't ever worry, like, what am I supposed to write? You're supposed to write nothing. You're literally just setting this up for the lovely day that you decide that you're actually going to run this experiment. And then we need to have, like, headings for this table. So bring this over here, and I'm going to say up here, this is the concentration of atrazine. Now these are my different trials, but trials for what? For the rate of photosynthesis. And then we can't just put that, we need to put the unit. Like, so what are we measuring the rate with? How do you do that? We're gonna do bubbles of oxygen per minute. This would need to be put in its little heading. So it's gonna look something like that. Obviously with some beautiful ruled lines. That's what it would look like, and that's kind of what we need to make. If you want to include an average, you can include an average over here. If you want to say like the mean. You could elongate it and do that if you wanted to. You don't have to. If you weren't counting bubbles per minute, then you were doing like the concentration of oxygen per minute, then that would be what you put for your unit. All right, without measuring this out, I'll do one really quick. Cool. Oh no, it's even more crooked. My gosh. Don't we just want to be done with exams and all the jazz? It is a little crooked. I'm just going to live with it. Somehow I'm going to move on with my life. All right, and we don't want to be like squeezing words in because then it looks like we have poor planning. This is something we're definitely going to do with the pencil as well if you're doing a data table. We don't want to be squeezing words in. So in real life, if I was doing this, I would write out concentration of atrazine first, and then where I have that comfortably fit, then I would put in this line. Okay, and then do something like this. Now up here I need to do the rate of photosynthesis. But when I did this like handwritten, I did have to erase, and then I did even use some whiteout. But you can't have that. Moles of oxygen per minute. And then I need one more line for my trials. And I don't know if I'm going to get this all to fit really nicely. But I won't go back and measure, it's fine. One, two, three, four, five. It's so big. All right. And if you're like, why do you have 6 there now? Because I made this space too big. But if I was using a ruler, which you absolutely should, it's a little crooked, I would definitely make sure it was spaced out enough that 5 fit comfortably. And it doesn't look like I have this big gap. Or if you have that big gap, erase those lines. You won't need them. And this, I'm going to call it the mean, because now I have this ridiculous base. Okay, let's actually pull it out. All right, there's 0. Okay, what do I have? There's 0. All right, this is the space I have. So I have about 5. So cool, awesome. I need to include I know I have 5 independent variables, but I also have my control, the 0. Okay, so it needs to be like 0.8 to make it all fit. So, here. That make a little line cute. And 2.4. Oh, no way. I'll fix it. Okay. I mean, that'll work. Good enough for YouTube. And now I just need to put in the concentration of atrazine. 0%. This is the control. I'm going to do it right. 0.2%. And you don't need to include the percentages or even the control, like labeling that as the control. That's kind of elementary. You wouldn't need to do that. And you do not need to put the actual percentage sign there. You could, and you probably should, put it right here. There you go. Now you don't have any data to put in because you haven't actually done this, but the day that your hopes and dreams come true and you decide to, like, I'm actually going to do this, how does atrazine affect the rate of photosynthesis? Then hooray. So that's what that looks like. This is going to be the longest thing that we do. And we're done with that now. And then we could say plot a graph, line graph with, so you could write this out or you could draw this out. Rate of photosynthesis on the y-axis and the concentration of atrazine on the x. Or you could literally just draw it out. Cool, love it. And this is going to be, but you need to have the units there. Finally, safety. Okay, they said in the very beginning, atrazine is toxic. So we don't want to say atrazine is toxic, period. Like, that is a statement that it's not a safety protocol. You don't want to be vague now and say, wear eye protection, wear gloves. That's vague. Same is wear personal protective equipment or even writing PPE. Like, okay, am I wearing a hazmat suit? Do I need to wear a certain type of like rubber shoes? Like, it's very vague and it's not specific. So we got to consider ways that we might hurt ourselves. And if you're wearing any sort of safety gear, you need to mention like maybe a reason why. So the atrazine is toxic. Wear gloves or goggles or use goggles. To prevent skin irritation or eye irritation or both. We have to use a light source so that we are able to maintain light intensity. And anytime you're using something that has a cord and maybe there's a liquid around, we're going to want to talk about keeping cords away from liquids. We don't want to say something vague like don't electrocute yourself. Like that's a good warning, but that's not a safety precaution. That's not a way to like prevent it. You're just saying don't do this. I'm going to actually squeeze it over here. Forgive me. Keep electrical cords, not the outlet. The outlet is what's like in the wall. The wall isn't going to hurt you. It's the cord. To prevent shock, prevent electrocution. Last thing, ethics. We are using something that's alive. We're using a plant. So things we should consider: where are we getting the plant from? What do we do when we're done with it? Is there a time that we don't want to maybe get this plant? Is there a certain type of species we don't want to use, etc.? Is there, like in the cleanup, anything that could harm the environment? Those type of things. All right, so don't remove plants from protected areas, like an MPA. I'm just going to state this and not write it, but you also could also mention, sorry to say also twice, but that you should not remove plants that are a food source for an endangered species, or don't remove plants that are a nursery ground for protected species, or that are a nursery ground during breeding season. And then only return, maybe we would say the control group plants to their native environment, natural habitat, because it said it's toxic, we don't want the atrazine to now be in the environment because we've placed all the plants back there. But the control group that didn't get any should be okay. And then don't wash atrazine down sinks that could lead to waterways. Oh, goodness gracious. All right, this was a big one. But that's the paper for October, November 2023.